All right, we're going to go ahead and get started and just move things along. So very pleased to be introducing Dr. Banerjee to you. I met him quite a while ago when he was in Dr. Orange's lab. And so he's really been pursuing not just high parameter flow cytometry, but the NK cells specifically. So it'll be an exciting talk and you'll get to learn a lot of what he knows about that. So Dr. Banerjee is an immunologist and assistant professor at the University of Texas, MD Anderson Cancer Center. You can see he's in Katie Rezvani's lab. He has more than 25 years of experience in cancer immunology and therapeutic development. As director of research for the cell therapy platform, they lead scientific strategy, benchmarking, and infrastructure development for advancing cell therapy programs. Their work has contributed to over 70 peer-reviewed publications, multiple patents, and successful partnerships with academic and industry collaborators. With over a decade of leadership in scientific administration, Dr. Banerjee has overseen multidisciplinary teams, complex research operations, and multi-million-dollar laboratory initiatives. They've supported numerous clinical trials through experimental design, data integration, and cross-departmental coordination. Prior to joining MD Anderson, Dr. Banerjee established the Center for Human Immunobiology right here in the medical center at Texas Children's Hospital, and he led major research facility design efforts in both academic and industry settings. He also serves as a subject matter expert and consultant to international scientific groups.
Thank you, Marcia, for this very wonderful introduction. And I also thank the organizer for giving me an opportunity to present my work over here. So basically today we are talking about the NK cell synapse. Now, we are trying to bring this concept from bench to bedside, and then it is going back to the bench side again. And regarding that, I must thank my mentor, Dr. Katie Rezvani, for his pioneering work in the CAR NK cell therapy. And with that interaction, I would like to move forward. So these are my little engagements with other companies. And as far as immunological synapse is concerned, if we ask a question to ourselves, what is immunological synapse? Then basically it is the interface where the effector cell on the top actually over here, it engages the target cell at the where the
Try pointing right there to that screen.
it's not going. So where the effector cell on the top actually engages the target cell at the bottom.
So they can see it online.
Yeah. Give me 1 minute, please. The mouse, fine.
Yeah.
Okay. So over here, it is the YTS cell, which is basically the human NK cell derived from a human NK cell line. And at the bottom is modified K562 cells with the CD86 gene in it. That's why we call it KT86. And as you see, the interface between the effector cell and target cell is right over here. So how it is helping the NK cell, this forming of immune synapse. Now, NK cell, they basically do two types of function. Number one, killing of the tumor cells, and number two, cytokine secretion. Now, for killing of the tumor cell, what the NK cell need, they have to attach strongly to the tumor cells. Now, for that, actually, we can see polymerization of effectin is happening at the cell-cell interaction, which is kind of, you can call a liquid cement that is being transformed to a solid cement and holding the tumor cell and effector cell together. And once that happens, then MTOC, microtubule organizing center, which is defined over here by alpha-tubulin staining, comes to the cell-cell interaction zone. Now, what is not being seen over here, that surrounding this microtubule organization center, there are lytic granules, which we call perforin. So perforin actually comes to the immune synapse, goes through it, forms tiny pores, as we defined by its name, and then through which granzyme comes and kills this particular target cell. Now, keeping that in mind, we thought of developing a concept of immune synapse to our trial. For that, we collaborated with a company known as Affimed. So that particular company helped us to develop a clinical trial against Hodgkin and non-Hodgkin lymphoma. So the cells from Hodgkin and non-Hodgkin lymphoma, the NK cells from the body of this patient, they don't kill the tumor. So what we did over here, we took the NK cells from cord blood cells and leveled those NK cells with AFM13. What is AFM13? It binds to CD16 on one hand and also stretches its arm to bind the CD30 antigen on the Hodgkin and non-Hodgkin lymphoma cells. Now, as a result, we can see that these two cells are forming a strong immunological synapse over here, where the accumulation of AFM13 is there, CD16 is there, and along with CD30. Now, keeping this concept in mind, we moved forward for our clinical trial, and here is a little schema for our clinical trial, where we actually took the cord blood derived NK cells, and this cord blood derived NK cells, we pre-activated them with cytokines and then expanded them for 7 days. And when the cells were being expanded, we also performed chemotherapy to the body of the patient. This is basically lymphodepletion therapy to the body of the patient, so that this patient will not be having any kind of cells to react with these NK cells, and the NK cell will be easily accepted in the body of the patient. Now, once that happened, then actually we infused NK cells to these patients. And we knew while we were infusing these NK cells to the body of the patients that there would be human leukocyte antigen mismatch between the cord blood NK cells and the patients, because it is very, very difficult to find 100% match. However, we took this particular phenomena in our advantage in order to find out if there is any circulating donor NK cell in the blood of the patient. For this, we did a simple flow cytometry experiment, and where you can see these are the smaller size of cells which I gated on. This is the singlets, this is the live dead, and then I gated on CD45 positive cells, got rid of the CD33 and CD14 positive cells, and from these lymphocytes, actually I split that particular group to CD56 positive and CD3 negative population. And once I did that, then I tried to see whether there is any circulating donor NK cell in the blood of the patient's body by staining the entire tube with human leukocyte antigen. Now, I was able to use, say, 6 different HLA antibodies to distinguish the donor and patient NK cells. I did it so many times, I can remember it. It is A2, HLA-A2, HLA-A3, HLA-B7, HLA-B8, HLA-BW6, and HLA-BW4. These are the 6 different antibodies that worked in my hand for flow. And request to you all, if you can find a good HLA-A1 antibody, let me know. I'd be very much interested to test it because I am HLA-A1 positive. Now, with that concept in mind, we saw, okay, there are donor NK cells in the body of the patient. So you can tell me, you just infuse the cells and after 3 hours post-infusion, you tried to see the cells. Yes, true. But we also found that when we infuse the cells, these donor NK cells, to the body of the patient, it persisted till day 14 to day 21. And of course, upon infusion, the peak goes up around day 7 to day 10. And then generally this peak actually comes down. Now, this particular persistence of donor NK cell that we found is basically not so much dose dependent, because when we infuse the patients with 10 to the power 6 donor NK cells per kg of the body weight of the patient. Then you can see it persisted till day 14. Same is true for 10 to the power 7 donor NK cell per kg of the body of the patient. However, for 10 to the power 8 cells per kg of the body of the patient, it persisted a little longer. So in this particular clinical trial, we were allowed to do up to 4 infusions. And since 10 to the power 8 cells per kg of the body of the patient did not give us any toxicity, we moved forward for 4 infusions with this particular dose for the donor NK cells, and we did not see any toxicity. However, it is easy to imagine, well, we are infusing the cells in the blood, and that's why you are persisting in the blood. And that's why I got a very limited opportunity to see whether these NK cells are going to the tumor site or not, or basically to the lymph node or not, where the disease is actually there. And the answer is yes. So this is the data from the lymph node of a given patient, where we see that around day 3 to day 4 and sometimes to day 7 of infusion, there are donor NK cells in the lymph node of the patient. And as we will be seeing a little bit later, that we actually used or got some 40 plus patients, but only 10% of those patients actually I was able to work with the lymph node of them. Why? Because most of them, the lymph node was calcified, the lymph node was not available, patient did not agree. So there are so many so forth things were there. However, you can see that more or less these donor NK cells were able to reach the lymph node of the patient. And were they functional? Yes. So this is a very complex slide, and I'll try to clarify it to the best of my knowledge. So first of all, on the left-hand side, we see how many cycles of donor NK cells the patient got, 2 and 4. As you can see over here, under division was 50-50. And then this is the number of patients, 1 to 42. And over here, the blue lines basically shows that response or ongoing response. And the gray line over here shows that the patient got partial remission. And of course, after a certain point, when you see the red dots, then actually we see that non-responder or the progressive disease had occurred. So from the very beginning, if we see that, we will see that from out of these 42 patients, basically 3 patients over here, here, and here were non-responders. And after a while, the progressive disease occurred in most of these patients. And we're trying to find a remedy for that, and I'm going to discuss it in the next couple of slides. So our donor NK cells are working, we can say, as far the response is concerned. But a question came to my mind, what about the patient NK cells? Whether the patient NK cells can also be functional, or we can make them functional for this particular disease. So now, if we just look at the CD16 staining, now this is the baseline of the patient NK cell, where we see that there are a lot of patient NK cells are over there. Now upon lymphodepletion, the number of patient NK cells came down. And of course, the red defines the intensity of the staining, it also came down. Now with time progression, we see that these patient NK cells are coming back along with the expression of CD16 on them. However, this is an infused product. However, for the donor NK cell, the situation is completely opposite. That in the beginning, we saw a lot of donor NK cell expressing CD16, and at a later on, actually, we see the number of donor NK cells are coming down and down. Now, there is something very important which I have to recapitulate over here. And as we have seen in the schema, that we are all, along with the donor NK cell over here, we are also infusing the patient with AFM13 antibody. And this infusion is occurring at day 7, 14, and 21. So what this AFM30 antibody is doing, this AFM13 antibody is also binding to the patient NK cells and also to the donor NK cell as we may expect. Because what we were doing, because before the infusion of the donor NK cells, we were taking those NK cells, then pre-labeling them with AFM13, and then infusing back them to the patient. Oh, good. So, so now again, we have the question in our mind that, well, we see the binding of the AFM13 in patient NK cell. How long do they persist? So the patient, this is the total infusion product where we say 80% of the NK cell are positive for AFM13. However, for donor NK cell, we see that the binding is around 20-30%, stays okay up to day 21, and then it goes down. For patient NK cell, since over here the donor NK cell number comes down on day 14, around day 14 to day 21, as I was showing in the previous slide, we see that a lot of patient NK cells are binding to this AFM13. Now, again, the binding of AFM13 to the patient NK cell does come down because after certain time, we stop injecting AFM13 to the patient cells. And also, you have to remember that the donor NK cells are dividing not that much as patient NK cells are. So patient NK cells are basically overtaking the number of donor NK cells in the body of the patient. And that's why probably it is coming down over here. Nonetheless, if you ask me the question whether this is functional, fortunately, I didn't have to do this experiment because in 2013, Reynolds et al. Actually showed this particular data that when they took the NK cell before therapy, before AFM13 injection, basically, these cells over here, these cells were not able to kill the target cell, which is L428, CD30 positive. And over here, actually, on the x-axis, they put the effector to target cell ratio in a decreased concentration. And on the y-axis is the percent of cytotoxicity in the increasing way as mentioned in this particular scale. However, upon NK cell therapy, they saw that after 24 hours, if they take out the cell from the body of the patient, and then it is able to kill the tumor cells. However, they didn't mention much about the function of AFM13 in this particular paper. They said that, well, AFM13 binding to the NK cell is increasing NKG2D on NK cell, which is true, which we also saw. It is increasing CD69 on NK cell, which is an activation marker, which we also saw. And that actually kind of backfired to us because the reviewer asked me a question, then show me what AFM13 is actually doing, whether it is really binding to CD30 on the tumor cells, whether it is really binding CD16 on the NK cells or not. So imaging flow cytometry came to my rescue. And over here, as we can see, that on the top, there is an NK cell, and on the bottom, there is a corpus cell, which is CD30 positive. And CD56 is the marker of NK cell, as you all know. So CD16 and AFM13 both are coming to the synapse. And indeed, the AFM13 is binding to the CD16 and as well as the CD30, which is already there on the surrounding of the corpus cells. Now, upon this very binding, what happens, as I was saying earlier, that effectin, filamentous actin, comes at the immunological synapse, which brings the perforin at the immunological synapse, and accumulation of effectin and perforin at the immunological synapse is a surrogate of NK cell cytotoxicity. And indeed, we have seen that too, that when we took the NK cells and we labeled these NK cells with AFM13, whether it was washed or was not washed, it didn't really matter. It was able to kill the corpus cells at 10:1 effector to target cell ratio. And this very binding of AFM13 on NK cell persisted up to 72 hours. And we saw statistically significant killing of AFM13 level NK cells over the non-binded or unloaded NK cells with AFM13. And also, this induced cytokine secretion of the NK cells, as we can see over here, interferon gamma, and TNF alpha, and indeed some other cytokines too. So right now, we can see from this data that we can basically induce NK cell cytotoxicity by binding the CD16 on it, or we can basically generate a card which is expressing CD16 on it. Now, why overexpression of CD16 needed? Because CD16 sometimes it shades off. If you overexpress CD16, then it will be persisting on the NK cell a little more, and the concept was that it can work a little bit better. With that particular idea, in the lab now we developed 3 musketeers. So this is our NK cell, and last time our NK cell did not survive more than day 10 to day 21. So this time actually what we did, we put IL-15 in the CAR construct, which will help the survival of the NK cell longer. Overexpressed CD16 on them, and side by side, we also put CD3 complex on the top of the NK cell, which can be expressed on the top of the NK cell along with the ITCR. Now, here is what happens when we try to figure out whether these NK cells that we generate will be functional or not. So I took the non-transduced NK cell on one hand and the plural NK cells or pluralink NK cells on the other hand. And then I labeled these NK cells with one particular anti-human antibody and tried to come up with anti-human IgG, which was linked to which was linked to Alexaflor 647 to see the very binding of the previous antibody on the anti-NK cell. So now, as you can see that the anti-NK cell, they express CD16, and that's why there is some binding of this antibody on the surface of NK cell. How do I know it is the surface of NK cell? Because I developed 2 masks, one on the outer surface of NK cell, and the other is the inner cell mask, which actually excludes the outer cell mask. So then I asked a question to the IDEA software of ImageStream, well, show me the data where these particular pink dots are colocalized with the outer cell mask, but not with the inner cell mask. And that's how actually this particular line is generated. Now, at a glance, you can see that the anti-NK cells are binding lot less antibody than the plural NK cells because they are expressing a way more number of CD16 molecules on them, which was quite expected. Now, apart from binding of the antibody on the NK cells, these pluralink NK cells have also other function. Now, suppose this is the pluralink NK cells. As we said that it can express CD3 on it. Fine. Now, all these T-cell engagers basically are FDA-approved antibody that we can use in our therapy so that these antibodies will bind the CD3 with one arm on the NK cell and will engage the cancer cell with its other arm. And indeed, the CD16 expression will help us in 2 different ways. Number one, it will increase the ADCC. And also, we can basically use antibody drug conjugate over here. We'll put the antibody drug conjugate in the blood of the patient. So the antibody will go and bind to the cancer cell, and the FC portion of the antibody will bind to the NK cell. So this antibody drug conjugate will kill the cancer cells, and also the NK cell will get one or another way to get close to the cancer cells. And we all know that some APC or cancer cell can present some non-MHC, non-classical MHC-derived antigen through CD1D, which the ITCR will recognize. And indeed, the NK cell, they have the cancer killing mechanism by the expression of NKG2D and DNAM1, TRAIL, FAS, etc. So it can kill the cancer cell that way. And as I was saying, that we have IL-15 in these NK cells so that it will survive longer. And indeed, these NK cells over here will also secrete some other cytokine as before, which will kill the tumor cells. So keeping that in mind, I did one experiment where actually I took these pleural NK cells and then labeled these NK cells with an antibody known as blinatumomab. So this is the antibody which binds the CD3 on this NK cell, and also it will bind the CD19 of the NK cells, the Raji cells or the malignant B cells. That was the whole idea. And then we took these malignant B cells and then we labeled those cells with obinutuzumab. So basically it will bind to CD20, but this Fc receptor will remain open. And this is the CD16 on the NK cell, which is not yet engaged. Now, while these two types of cells were conjugating to each other in a test tube, I also added alpha-gal to wanted to see whether these cells in an ideal scenario, by engaging all these killing mechanisms, can be functional. And indeed, when I see these NK cells on the top and a malignant B cell on the bottom, I see that CD16 came at the synapse. CD56 was already there. Now, if you look at CD19, and at the faint bottom over here also shows that this is the B cell, and the accumulation of CD19 at the synapse is not what we not expected, because we are using an antibody which can bind CD3 and CD19 together. That's why both of the molecules are at the synapse. Then ITCR is at the synapse as well, along with perforin and effectin. So basically, I can say by in vitro experiments, these NK cells are functioning. One most important point over here is that this CD16, ITCR, and CD3 complex are expressed on various localization on the surface of the NK cell. However, only 20 minutes of conjugation with tumor cells, all these three things have come at the junction of tumor cell and NK cell, which we call the immune synapse. So that's how we can say that when we studied the AFM13 labeled tumor cells, then we saw CD16, CD30, effectin, and perforin are coming over here. But over here, when we studied these cells, then we saw that CD16, CD3, CD19, ITCR, effectin, and perforin coming over here. So the strength of forming of immunological synapse is way higher in these pleural NK cells and in comparison to this AFM13 level NK cells. And by using imaging flow cytometry, we can also not only measure the strength of immunological synapse, but we can also measure what are the signaling molecules and how the synapse is forming. Say, for example, LFA1 is beta-2 integrin. It comes first at the synapse, then comes effectin, then comes perforin. So stepwise formation of immune synapse we can also learn. However, there was a caveat in my mind that, well, if I generate a graph and show that, okay, these cells are forming synapse, these cells are bringing effectin at the synapse, then somebody might ask me whether this is the same conjugate is bringing the effectin, perforin, and other molecule at the synapse. So in order to answer that question, I came up with this idea. It is a merge of the two different tools. One on the top, we see the IDEA software, by which I picked up the conjugates over here, which is basically depending on the aspect ratio and the area of the cells, then the focus cells, then the live cells, then on the y-axis it is CD30, on the x-axis is CD56. So the cells which are both positive are my conjugates. Then I took these particular conjugates and split it in different Disney plots. The first Disney plot is showing the total synapse area of all these conjugates. So as you can see that some of these conjugates are forming strong immune synapse as defined by red, and some of these conjugates have lower synaptic area. But in some of those conjugates which has that higher synaptic area, also bringing LFA1 at the synapse, but effectin is coming in some of those LFA1 covered area, and then perforin is coming probably both the areas which is covered by effectin and LFA1. So taking together, this is the overlap of everything where you can say, okay, if I have 100 conjugates, out of this 100 conjugates, only 20 are forming strong immune synapse, which could be functional in our therapy. And indeed, we can compare groups versus groups of NK cell. Over here, the NK cells are different, but I kept the tumor cell the same. And I can see over here that when the immunological synapse is formed between this CAR-NK1 versus CAR-NK2 with the same tumor cell, then we can see that for the CAR-NK1 tumor conjugates are quite stronger than the CAR-NK2 tumor conjugates. Now, remember that when we develop our CAR-NK cell, they are cytokine stimulated. So visually, it might not be very easy to see the difference, but if we see the statistical data, then we will see that more number of conjugates are having everything at the synapse. Now, with that hope, actually, we started our NK cell therapy with this pluripotent receptor for B cell autoimmune disorders such as SLE and to some extent rheumatoid arthritis and multiple sclerosis. Hope to see a good immune synapse formation there where our NK cells are killing the tumor cells. And I would like to thank everybody from the Dr. Rezvani's lab, especially Katie and all these other people, and my undergraduate student, Benjamin Shah, which is sitting over there, Majid, Sanjida, and Islam, they are also very helpful to me. And special thanks to family, patients, and physicians. So I'll take questions now.
I'll always get it started. What's your favorite thing about the NK cell? You've been studying it for a long time. It's really exciting to see that you're able to do this work with immune synapse, especially because I would say anywhere between 50 and 80% of therapies are targeting, proteins in that area. But the question is, what's your favorite thing about the NK cell? And then, where do you want to see this work go next?
Yeah, actually, I am studying NK cell synapse. I didn't put those slides over here because those were not from imaging flow cytometry, but previously joining to Katie's lab, I used to study WAS, Wiskott-Aldrich syndrome, and I used to screen those patients by immunological synapse formation. From that, actually, I'm developing the area over here because WAS patient has everything, but without the functional WAS gene, the effectin does not come at the synapse. So I was also thinking that, okay, since I know that that happens for a therapy, and then Dr. Orange treated those patients with IL-2, which actually overcome the WAS-dependent polymerase of effectin at the synapse by OF2. So I was also thinking that, hey, if I could study the immune synapse formation with my CAR-NK cells, probably I'll be in a position to pick up the better CAR-NK cell for the therapy.
And then just to follow up on that really nice example of, a use of imaging cytometry, what kind of, probes or reagents are missing to better understand the immune synapse, right? Because we don't really have a lot of probes that can get into that inner cortex or above the actin cortex, that inner leaflet. Do you have any idea what would be needed, right? They'd be fluorescent probes or... you did a really nice job because you're creating that mask, those two different masks, but I just didn't know if you know anything coming or anything that's out there to help us improve being able to really
Yeah.
characterize.
That is one of the concepts with this plural NK cells, that if we put many things that can attack the tumor cells, maybe everything will come at the immune synapse. But the question is how long they can stay, and whether, the NK cells are also known for serial killing activities, whether we are forming too high of a concentrated immune synapse so that they cannot detach and kill other cells. So these are the areas that we are still developing right now.
Okay, thank you.
Thank you.